Welcome back to The Report. Tonight, our Age of Design series continues. We're looking past the parts on the table and asking whether there's order and timing that all makes life run, right? If we have all of the elements for life all laid out in front of us, how do you make it turn into life itself? Dr. James Tour will be joining us as we move from the plan to the practice. How does a cell take the DNA code, organize the work, power it, and protect it so it doesn't even fall apart? So basically, if you have a fully functioning strand of DNA, RNA, you still need many other molecules for life to work. In fact, life depends on 4 classes of molecules working together: nucleic acids, DNA, RNA, which we just discussed, stores and transmits genetic information. You've got carbohydrates providing energy. You've got proteins made of amino acids performing so many different functions, especially as enzymes. Then you've got the lipids that form protective membranes that enclose and organize cells. And those aren't just important molecules. They're essential ones. So without all 4, life as we know it cannot exist. Dr. James Tour is back with us to dig deeper into this topic. Dr. Tour, what do you mean when you say that even the building blocks of the building blocks haven't been made?
All right, so the building blocks are the 4 classes of compounds, the polymeric structures of the compounds that you've just outlined. So to have proteins, you have to first have amino acids. So you have to be able to make 20 different amino acids and have them all chiral, all handed. Of the 20, 19 of them have to be chiral. So one of them doesn't have a chiral center, it doesn't have a stereogenic center. So all of them have to have an anti-purity, 19 of the 20. So those are the building blocks of the building blocks.
Okay.
If you want to make polysaccharides, the sugars, the polysaccharide molecules, you have to be able to make first the basic sugars before you can hook them together in the polysaccharide. Before you can make a polynucleic acid, DNA or RNA, you have to have the nucleic acid, which has to be solved. And in order to make a lipid, you have to have a glycerol and a fatty acid and a glycerol. And we don't know how to get those fatty acids pure. We don't know how to get a stereogenic center on the fatty acids, and we don't know how to distinguish what's called the enantiotopic ends of a glycerol to get these things to be chiral. So we don't even know how to make the tiny pieces that make up those 4 classes of molecules.
Wow, okay. So what do you think is if you had to pick one, the toughest chemical challenge that we currently have in origin of life research?
Well, there's many of them, but one of the tough ones is the synthesis of the sugars and hooking them together. How do you hook the sugars together? So if you just have 6 glucose molecules, you have to, there's 1.2 trillion ways you can hook together just 6. So when you have hundreds of them hooked together, and they have to be hooked together in the right order or the cell doesn't function, that's a real problem. How do you do that?
Yeah.
So that's, again, just one of many. It's hard to distinguish which one is harder than the other because every one of them is hard.
Right. When you have these overwhelming obstacles and you could just pick one, and I mean, you could talk about it forever on any one of those obstacles that have to be leaped in order to solve this problem from a natural perspective. But then again, we see these headlines, we see them on the news, we see them on the science websites, we pick up a magazine and we're flipping through it and it's like, oh, we figured all of this out. Why do you think that progress in this particular field is often overstated in general media?
That's an interesting question. It is highly overstated. And the researchers know that it's highly overstated. They know they're not just on the verge of figuring this thing out. I am not sure why they keep saying this. Is it because they think they have to say this in order to keep their grants flowing, in order to get money?
Yeah.
I don't know. Is it something more philosophical, something deeper here, that we have to suggest that we know the origin of life, and we know that it has this materialistic bent to it, so that we can somehow exclude some higher being that has done this. I don't know why they permit this to go on, that they're just on the verge of this, but I can tell you that they keep dying of old age without ever having solved this. So decade after decade, they keep saying they're very close, and they keep dying. And they're going to continue to die of old age before they ever solve this problem. And then the next generation will say they're just on the verge, and they'll die too, and the next generation after that, because we're that far away from this.
You know, Dr. Tor, I think that's a great point. And as a Christian, I read something like 1 Samuel 2 that says that the Lord brings death and makes alive. He can make people alive. He brings down to the grave and he raises us up. And so when I see that there is, I believe that there's a divine being that created us all with purpose, that we are wonderfully made, that we're made in his image. And that puts an entirely different meaning to life and the origin of life issue and how we can fully understand it. But when it comes to the different processes, the different classes that life depends on, I want to get your opinion on a couple of these. Let's talk about carbohydrates for a moment. The molecules expected to provide both energy and structural support. And this is where chemistry gets real. This is way beyond a lot of things that I might even be able to comprehend. Sugars like ribose struggle with instability in water, messy mixtures, unhelpful side reactions. I want to step into carbohydrates for a moment and see why even the very first rung on this ladder of the natural origin of life is so hard to climb. So talk about carbohydrates for a moment and explain on a layman's level how we can understand these things and what the issues are if we're trying to answer this question.
Okay. Well, carbohydrates are sugars, the very problem I was just talking about. What happens is carbohydrates will have multiple sites where there's what's called a chiral or stereogenic center. These can all have a hand in this, every one of these centers. And so since you have so many of them, you can have 5 centers, or you can have 4 centers, depending on which particular carbohydrate you're talking about. And so you get vast mixtures, because you get on the ones that have 5 centers, you have 2 to the 5th possible isomers, and the ones that have 4, you have 2 to the 4 centers. The ones that have 3, you have 2 to the 3rd possible center, so you might have 8 possible isomers. And these compound the problem because you have so many different possible isomers, meaning that they're similar in structure, but they're not the same, and only one of those is going to work. And if you have mixtures of them, that gums up the work and it doesn't function. The other thing is, how are these two going to hook together? Say, I can hook thumbs to thumbs. Yeah. I can hook thumb to forefinger, thumb to middle finger, thumb to ring finger, thumb to pinky finger. So there's all these different combinations. And then on the next molecule that it hooks to, again, are all these combinations. That compounds the problem of how you hook these things together. Number one, what is the chemistry that you use to hook them together? Nobody knows what the chemistry is on a prebiotic Earth, because this is where I talk about the free energy is positive. They favor not being together. They don't want to be together. So how do you get them to hook together? Well, in nature, you use enzymes. In the lab, we have these clever techniques that we've figured out. But even with our clever techniques, it's very hard to hook the right one to the right. And so what we do is we block all the other sites and we just hook to the ones that we want to. It's a lot of laborious chemistry. Big problem to solve that. And then we can't hook them together. So we can't make these little things without getting vast mixtures, and we can't hook them together without getting vast mixtures. So this is the genesis of the problem with carbohydrates. It is the most difficult class of compounds. This is and if you have, and then plus carbohydrates can have they can have a hydroxyl group on the end, which is called the anomeric center, can be up or down. If it's in one configuration, people can, for example, eat it, and you're eating You're eating some carbohydrate that you can digest, for example, wheat. But if you have the other configuration, then it's a carbohydrate that we can't eat. That's grass, for example. Our bodies cannot digest that. And that's just a little varying of the hydroxyl group going up or down. Depends on whether we can even use it for food, let alone use it for other biological functions. So this is the problem. If we eat it if we eat grass, for example, it goes right through us. It's called fiber. It goes right through us, but our body doesn't get any content to be able to live off of from that. It might be good for our digestive system, but it's not good for much else in giving us energy. So that's the inherent problem with carbohydrates. That's just one of the 4 classes of compounds that if you don't get it right, it doesn't work. And it's not like you can mess around with this thing. This has to be right. Yeah. None of these problems have been solved with carbohydrates.
And again, I love it, Dr. Tour, where you mention the complexity of all of this. And if you get the chirality of something wrong, it doesn't work. If you flip it on its head, it doesn't work. If it doesn't connect up, or if it creates a mixture instead of the exact right thing, it doesn't work. Again, we understand this now within chemistry, but nature doesn't have a mind. Nature can't possibly comprehend all of this and somehow will it into existence. So even if we could clear the sugar hurdles, life still needs workers, right? That means protein. So let's move for a moment from energy and scaffolding to the active machinery inside the cell. Proteins are a sequence of amino acids. You mentioned that we see 20 standard types that need to be specifically arranged and then folded into chains, sometimes 500 amino acids, right? But these chains are also working with other chains to form enzymes or molecular machines with sometimes thousands, 5, 6, 7,000 of these amino acids put together. And a lot of origin of life experiments claim that amino acids can simply form under prebiotic conditions. What do you see as the major challenges with those claims?
Right. So if you use the Miller-Urey experiment, that made about, I don't know, 12 or so, 13 or so of the amino acids, of the canonical amino acids that you need of the 20. But what it did is it took the simplest of the amino acids, glycine, and it made a very large amount of that. And then it took the next simplest and it made a lot of that, to the exclusion of all the others. And you can't get a functioning enzyme that's made predominantly out of this simplest glycine, and these would dominate. The other problem here is that these amino acids react with the carbohydrates. So you had to have kept the carbohydrates far away from this, because they would react with what's called a reducing sugar. All of these carbohydrates have reducing sugars. They have an aldehyde on the end. And these amino acids have an amine. That's why they're called amino acids. And those amines will form what's called an imine with a carbohydrate. And then you get dehydration and you get this Maillard reaction and you get this decomposition. And everything starts forming these polymers which are unusable. So in any origin of life scenario, you have to say, well, somehow I'm going to make my amino acids and keep them totally away from other molecule classes where there's a carbohydrate. And how do you do that? And then all of a sudden, when you want there to be carbohydrates, you got to somehow introduce it. How did that happen on an early Earth? Nobody knows. And then the amino acids, half of them have a stereogenic or chiral center. Yeah. And I'm sorry, 19 of the 20 have a stereogenic or chiral center. How do you control those? Nobody knows. And about half of them have what's called an active side chain, meaning that they can hook up in a wrong order, in a wrong way to attach. So half of them are having this wrong attachment system. So it's very hard, and you have to have these enzymes that construct us. And that's why we might eat a bagel for breakfast this morning, and by lunchtime, that bagel has been broken up into pieces and is part of one of the cells in our ear, for example. How did that happen? It's because enzymes, these protein-based systems, most enzymes are are made up, are proteins made up of these polypeptides. And they are the little machines that will build us, that will construct us, will take molecules and set them in certain places. And so you have to be able to have these and have these right. We have no idea how these things form. And plus, they have a code, they have an order that they have to be in order to be able to carry out the function that they need. It's not just a random sequence, it's a very specific sequence, which causes it to fold in a very specific manner that has an active site where that analyte molecule can go into and react with it. And then there's many enzymes that need coenzymes, that by themselves, they don't work alone. They have to work in concert with other proteins and form dimers or trimers or tetramers, like hemoglobin is a tetramer. And then other times they have to work with coenzymes, other small molecules that have to work in conjunction with them. And so to get these things to work, they need a lot of infrastructure around them. Nobody knows how all this came together. And remember, you can't have two lines of evolution going on, two lines of chemical changes going on in molecules that are disparate in the earth. These coenzymes have to be right there in place with the enzymes. And remember, these enzymes aren't going to last long. If you happen to have, just by randomness, if an enzyme happen to form, how does it fold up into the structure it needs to? Most of these need what are called chaperones, other enzymes to help them to fold properly. So in other words, if you have a hammock, and you say these hammocks that are made out of rope, how do you think that rope can fold into that hammock structure just by itself? No, somebody had to weave that rope into the hammock so that it's a hammock. You can't just leave it out in the wind and the rain and expect it's going to fold into this structure of a hammock. It doesn't do it. And you say, well, I'm taking a hard example. That folded structure in the hammock is really hard to do. No, the enzymes are harder. The enzymes have more combinations that they can go through. And so each one of these enzymes has to have a chaperone, another molecule that helps it to fold and to orient. So if you don't have the folded structure right, you just have a rope. You don't have a hammock. It had to have been tied properly with these in this ordered arrangement in order to have that rope now be a hammock. This is the same type of things that happens with these polypeptides. It has to now be able to fold into this structure in order to be usable. How does this happen? Nobody knows. And if you just say it folded by randomness, if it were just 100 amino acids long, there's not enough time in the universe, in the 14 billion years predicted for or estimated for there to be a universe, there's not enough time for that to have randomly tried the combinations to fold right. It would still be thrashing about today. But plus, it only remains in water for a matter of days. Yeah. And then it starts falling apart. And so that's one problem on top of another problem.
So we got probability is a huge problem here. You just walked us through how the Miller-Urey experiment is presented, even in much modern literature, as this success story of, oh, we've created life. But you've actually showed us how this is actually a real challenge, and you have to get things in the right order. All right, so let's just say that with all of those challenges, what if we have fully functioning amino acids set, and maybe they're somewhere out in the distant reaches of the universe, and a meteorite then delivers those fully functional amino acids to us? How does that work?
Yeah, so you have to get 20 amino acids, not just one. And they all come as mixtures, and they all come mixed with many, many other molecules. And so they generally don't stay around. And you have these amino acids, if they happen to be around a carbohydrate, they don't stick around. They form these Maillard reactions that I just mentioned. Now, let's say all 20 amino acids were delivered here on a meteorite. Yep. Now what? They don't want to hook together. Again, the free energy is positive. They'd rather be apart than together. So there's no mechanism to get them together. Now you might say, well, I'm going to use a wet-dry cycle. Yeah, they've made 14 mers, but they were never able to deal with that pendant group. which can also interfere. So you can't deal with dependent groups. And so they have to find a lot of other support molecules to be able to have clever coupling chemistry to be able to work. It's very difficult to envision that, very difficult to deal with this, especially if they have a side chain that bears an amine or a side chain that bears a carboxylic acid. These totally interfere and they'd become useless. So there's no way to solve these things. And origin of life people can't gloss over this with me.
All right.
YouTubers gloss over this with me because they don't understand the inherent problem. But no origin of life researcher will engage with me to discuss these problems because they know it's unsolvable. Yeah. They know it's unsolvable. So that they will not engage with me, that they will refuse to have these public discussions with me, is telling in itself. That's what I'm talking about. They will not have these discussions because they're organic chemists. They see exactly what I see. It's not that Jim Tour sees these problems and nobody else does. Everybody working in the community sees these problems, but they don't talk about it.
Yes.
And that's the problem.
That's a huge problem. And then you've got those on the outer edge who know just enough to be dangerous but don't fully understand the complexity. And they say, well, surely it's possible. We'll figure it out. Dr. Tour, you talked about probability being a huge issue here. I've got one more question when it comes to proteins. Talk a little bit about the you call it a chicken and egg, basically, paradox of proteins needing enzymes to form.
Right, so all proteins, the majority, not all, but the majority of proteins, certainly that are going to be sophisticated enzymes, need other chaperones, other enzymes that are going to help them to fold. Well, who caused the chaperone to fold? Another chaperone. Well, where did that chaperone come from? It had to have had another chaperone. So we don't know where these things, so we don't know where this inherent information came from for these to be able to form and fold. So all of that is an unknown. That's the chicken and egg problem. It's not been solved. And everybody who works in this area knows it's not been solved, but they don't bring it up.
It's incredible. You know, even a toolkit of working proteins can't function out in the open. It needs a protected, organized workspace that will then separate inside from outside, maintain the gradients in the right order, control traffic, and that, I guess, brings us to lipids. So we're talking about the foundation of the cell membrane forming a bilayer that separates the inside of a cell from its environment. And it's not really just like a passive wall, as I understand. It's a dynamic platform where other crucial molecules can integrate. And then proteins embed within the lipid bilayer to act as channels and pumps and receptors and basically allow the cell to control nutrient flow, waste removal, and even the signaling of a lot of that. And then carbohydrates attached to lipids and proteins on the surface, creating recognition sites that allows for communication. It also helps protect them. But what I'm saying is that lipids provide this structural framework that makes cellular life possible, but it works hand in hand with all of the other things that we've talked about so far. So, Dr. Tour, why are they considered the essential building blocks for life?
Well, let me first say, I'm impressed. I mean, you learned all of what you just said, which is really impressive. Most people don't know this, that if you're going to have a functioning lipid bilayer, you have to have them hooked up to carbohydrates, which provide recognition. There are also other proteins that are hanging off that provide recognition sites, and then you have protein channels, these transmembrane proteins, which are needed to have respiration. And then you also have other carbohydrate type structures within the lipid bilayer that act as ionophores, that act as mechanisms by which ions can flow in and out to keep the gradients, these transmembrane gradients operating. The lipids, in order to make lipids, you have to take a fatty acid, which are not that easy to make. Some people say they come out of hydrothermal vents. I'm not sure that on prebiotic Earth you had these things coming out of a hydrothermal vent, but even if you did, they've got to somehow react with glycine, which is 1,2,3 propane triol. And They have to be able to distinguish the two ends, which are identical, except to another chiral compound. And without enzymes being around, how do you pick out one and not the other? They're enantiotopic. It's not known. And then you have the two position being reacted as well. And these are esters. And so they want to hydrolyze. Esters hydrolyze in water. They don't want to form. Again, this is another positive delta G problem. So you have to somehow catalyze this, heat it up, and draw the water out as these are forming. You have to do something to push this reaction forward. They don't normally form, and they'll want to hydrolyze and go back. And then they've got to get in this bilayer structure, which can happen under shear. But just having a lipid bilayer structure, which is called a liposome, doesn't do it for you. You have to have the interaction with the proteins, which we just showed couldn't be made, and with the polysaccharides, which we said couldn't be made. So that's the problem with lipids.
Interesting.
And many of the lipids actually have an extra stereogenic center somewhere along that hydrocarbon chain.
Okay, so we actually talked about, and I won't go too much in the weeds here, but we've talked about chirality in different areas so far. Does that play a part in lipids as well?
Absolutely. That's what I'm talking about. There are enantiotopic ends to the glycerol, and that is all a chirality issue.
Interesting. Interesting. Okay. We've got so much more to cover, and I don't want to go too long here. So what I'm going to say is we've got basically components on the table, but now we need to shift our question from what life is made of to how actually the instructions for life are put together. Basically move from raw chemistry to now an extremely specified sequence, even though everything that you've talked about so far is highly ordered, it appears, but something that could be read and used. So let's talk about fact 3. Let's talk about DNA for a moment. Prebiotic forces can't arrange DNA nucleobases that contain the stored information of the DNA strand. So even if we've got a fully functioning strand of DNA or RNA, we've got carbohydrates, proteins, and a lipid bilayer, you still have to have this stored information to direct functions, right? To tell it what to do in the DNA code. And I'm talking about basically nucleotide bases, adenine, thymine, cytosine, guanine. In an extremely simplified fashion, it's like the blueprints, the recipe book for life. And those letters are pairing up to form chains. You mentioned a little while ago that a group of 3 of those letters, codons, basically can act like words that will tell the cell what to do. You put all of that in the right order, and somehow it all comes together. But that information has to be ordered. It's almost like a computer code. And then you think of the complexity of some of this, where the direction of protein development in human DNA, I think it's about 3 billion base pairs. But even the simplest cell, we're talking about thousands or hundreds of thousands, right?
Hundreds of thousands of what?
Of base pairs in just
yeah. Millions of base pairs for.
Okay.
And yes. Yeah, and in the simplest ones, yes, hundreds of thousands. So that's a real problem. So you said if you already have your DNA and RNA, if you already have your DNA and RNA, where'd the code come from? It's just a string of letters that are going to code for nothing useful. They code for nothing useful. So you had to have had information put in before you can have any useful DNA and RNA. Now you start with the nucleotides, and you have to polymerize the nucleotides together. Nobody knows how these go together because, again, the free energy is positive, which means these do not want to go. So you have to have some other clever methods where these can hook up. And they don't hook up cleanly. You get branching. They get, in the case of RNA, they get this 2' 5' linkage. In the case of RNA, they need a separate base. You exclude one of the bases on the DNA, you have to bring in uracil on the RNA, so it needs a separate base. The DNA can't make doesn't make proteins directly. It has to go through first templating to RNA to get RNA made, and then from the RNA, generally you get the proteins. That's the normal sequence that we look at. And you need lots of support enzymes. So how do You need enzymes to string these things together. You need a code to string these things together. Nobody knows where that came from. You need enzyme support for any one of these. DNA to be useful to make RNA, for RNA to be useful to make proteins. You need other support enzymes that bring in all the individual amino acids when you need them into place in order for this to happen. None of that is known. None of that is known how that happened on a prebiotic Earth. We are utterly lost on that when it comes to the origin of life.
See, this is something that I think our audiences need to know. The public needs to know these issues that are being faced that are not being talked about. I mean, Dr. Tour, you've been talking about them for a while, but very few people have tried to even mention the unspeakable of we don't know on this origin of life issue how it could have happened by itself. But every time that we're talking to someone, I always like to relate it to something that we could understand. Like, I mean, let's just talk about a written code, right? A book or something like that. How does DNA's code resemble a written work, like a book?
Oh yeah, and it's much longer than a book.
Okay.
I mean, it prescribes what RNA is going to be made, and then the RNA prescribes what the order of attachment is of those 20 amino acids to make the proteins. And it's that order of attachment that is going to define the function of that protein. Is that protein going to be used to make more RNA, or is that protein going to be used to break down the carbohydrates that you've just eaten, so that that energy can then be applied as glycogen in your body so that you can get energy from it. Enzymes do all of these functions, and the functions are defined by the order of the amino acids in it. So if you don't have the inherent order in the RNA, which came from the inherent order within the DNA, you're out of luck. I mean, it's just not going to do anything. It's just going to be a bunch of gibberish.
Well, you know, again, I would probably laugh at someone who handed me even a simplistic book or an article even and said, you know, that created itself. Books don't write themselves. So what do you say to students who believe that DNA could just have evolved its information?
Yeah, that makes no sense.
Yeah.
Only an idiot would say that. That's what I have to say. It makes absolutely no sense. It's really nonsense to say that DNA could inherently form with a functioning code. Things don't happen like that any more than you can take a random box of letters, shake it together, dump it on the ground, and have a written book. You might occasionally find a three-letter word.
Yeah.
On occasion, you might find that. But it's not next then to another intelligible word where you're going to form a book out of this thing. Right. And remember, even if you had 3 or 4 letters in a word, 3 or 4 words in a string that happen to form, it's not going to make any sense. There's nothing on either side of that. You need lots of information. So nobody in their right mind even says this except people who know nothing about the field.
Yeah, that makes perfect sense. On an intellectual level, we have to consider that. Put simply, just having the right pieces is not ever going to create a living system. It's always purposeful order. I mean, you can line up bolts or gears or wires or anything without an intentional plan, nothing's going to run. And that's the tension that we keep finding the deeper that we look at this issue. We've got one more part to this issue, this Age of Design series, where we want to take it just one step further and wrap it all up in a bow. We're going to pause here for just a moment, but we will be right back on the Genesis Science Report.